so it's great to have Dave Bartel with us. He received his Bachelor's of Arts at Goshen College in Indiana and then his PhD at Harvard University in Cambridge, and then became a Whitehead Fellow, and from there, assistant professor of biology at MIT, associate member at Whitehead, all the way up to being a full professor at MIT. It's wonderful to have Dave with us. Take it away. Sorry about all the interruptions.
Yeah, no problem. And thank you all for sticking with us. Sorry for those of you who, I guess, can't get in. Yeah. So my lab's interested in post-transcriptional gene regulation, why mRNAs from some genes are much more stable than others, some translated much better than others. And so, and we work on microRNAs as well as the mRNAs, poly A tails, UTRs that mediate this post-transcriptional gene regulation. And so today I want to tell you a story, a microRNA story, and also a messenger RNA story. So with respect to microRNAs, we all know that these are these small RNAs that get processed from these hairpins, loaded into Argonaut, and from there they downregulate messenger RNAs. And soon after we had found that there are lots, and others had also found that there are lots of these microRNAs, we found that each microRNA can have many targets. So there are about 90 highly conserved microRNAs from humans to fish. And these 90 families, on average, each have about 400 conserved targets in mammals. And so often these microRNAs will converge onto the same UTRs and regulate the same genes, but this still adds up to more than 60% of all the human mRNAs are conserved targets of microRNAs. And when we saw this, we realized it's going to be hard to find a disease or developmental process that isn't somehow influenced by microRNAs. And in fact, when others went in and knocked out either individual microRNAs that are highly conserved or multiple members of the same family, they found many different defects and development of lots of different places, depending upon where the microRNA is expressed, as well as cellular, physiological, and behavioral defects. And so often these are actually quite severe. So there's over 15 cases where 15 of these families where the knockout gives either an embryonic or neonatal lethality. And in other cases, you have later death or other severe phenotypes like blindness, deafness, infertility, cancer, epilepsy, et cetera. So these microRNAs are really playing really important roles in mammals. The way that they regulate so many targets is pairing to what we call the seed region of the microRNA, nucleotides 2 through 7, often supplemented by another pair here to nucleotide 8, or an A here across from nucleotide 1, or both. And this is the vast majority of targeting in mammals. Although there are a few cases where you have very extensive complementarity and you actually get slicing of the mRNA, but that's only been seen in 21 examples for all the microRNAs combined, 21 examples in human cells, whereas this is about 40,000 conserved sites. So we've been, quite interested in, how these microRNAs are made, their genomics, their evolution, how they get loaded, what happens to these targets once they engage their targets, how they recognize their targets, et cetera. But today I want to tell you another developing story in the lab, and that is the regulation of the microRNAs themselves at a post-transcriptional level. In other words, the stability of the microRNAs and what influences that stability. And for a long time, we've known that some microRNAs are really incredibly stable. So this is work from Eric Olson's lab where he showed that this microRNA, miR208, sticks around a week after the transcription's been turned off. Okay, so this has an incredibly long half-life, presumably through protection by incorporation and association with its ACO protein. On the other hand, there are some microRNAs that have shorter half-lives. Here's an early example from our lab. Olivia Rislin showed that miR503, quite a bit of it is gone only 12 hours after its transcription was shut down. So half-life just a few hours here for this microRNA. And Elena Kingston, a graduate student in the lab, has has looked at this systematically. She used metabolic labeling to generate half-life values for hundreds of different microRNAs in different cell lines. Here we have mouse embryonic fibroblasts, contact inhibited, and the half-lives are about median of about 34 hours. Okay, so well over a day for most microRNAs. And whereas the messenger RNAs in these cells have half-lives of only 2 to 3 hours. Okay, so the messenger RNAs are going to be much more responsive to some sort of perturbation, some signaling or other change because they have shorter half-lives, whereas the microRNAs are not going to be that nimble. Okay. On the other hand, it's the microRNAs that are helping to cause the messenger RNAs to have shorter half-lives, so they help to make the messenger RNAs more nimble. And of course, there are a few microRNAs here that have shorter half-lives. Okay, you know, about that of a typical messenger RNA. So what is it about these microRNAs that cause their half-lives to be so short compared to the others? And a clue to that came from work from Joan Steitz's lab, and as well as Phil Zamor's lab. So in these papers, well, first of all, Joan's lab showed that there was this non-coding RNA in one of the herpes viruses that directs the degradation of a host microRNA, miR27A. Okay, and you can see that here the pairing is not only to the seed, but also to the 3' region of the microRNA, quite extensively paired. Okay, and that's not what we usually see for microRNA targets. And she showed that in the presence of this non-coding RNA, HSCR1, that this microRNA is, you know, goes away quite quickly compared to when there is no viral RNA. Okay, so here's a viral RNA that is directing the degradation of this microRNA, and this is called target-directed microRNA degradation, or TDMD. And at the same time, the Zamor lab showed that you could use artificial targets with this extensive 3' pairing, and that could also cause some target degradation. And also they were observed that you would also have tailing and trimming of the microRNA, and that was thought to be somehow on the pathway to that degradation. So, and then more recently in McRae's lab, collaborating with the Stites lab, has solved the structure of some of these TDMD triggers with targets. This is an artificial one that pairs to miR122, and you can see the microRNA in the target, and what happens when you have this extensive pairing to the 3' end of the microRNA, that sort of causes a conformational change in the protein as well as the RNAs that releases the RNA from the pocket in the pause domain and makes it accessible. And that's the reason that you get these tailing and trimming activities on these extensively paired microRNAs. So our work on this began with the Cyrano long non-coding RNA. This is a long non-coding RNA that is conserved from fish. We actually found it in fish, but also had been annotated in humans and mice. And so it has these regions of quite extensive conservation, and that interested us, and especially when we saw that within this region here, there's this extensive complementarity to miR7. Okay, now the microRNA is on the bottom. Here's the 5' end, the seed pairing, and then you have 14 highly conserved base pairs to the microRNA here at the 3' end. Again, unlike anything we had really seen in the UTRs of the messenger RNAs. So that got us very interested, and Ben Cleveland, a postdoc, former postdoc in the lab, decided to look and see what would happen when he knocked out this region, pretty much knocked out the Cyrano link RNA in mice. And what Ben found, well, first of all, the mice were fine. You know, he didn't see any striking developmental or behavioral defects, but he saw this really striking molecular change where when he sequenced the small RNAs, this is looking at the fold changes in the knockout compared to the wild type. For the vast majority of RNAs, there's really no difference. But for the miR7s, and these come in 2 different flavors in mice, they went up about 50-fold in this brain tissue. And when Ben went on to look to see if there's any changes in the primary transcript or the pre-microRNA, or here in this experiment, you can see what's happening with the passenger strand. He didn't see any differences. Okay, so this increase in the microRNA appeared to be due to a stabilization of the microRNA that was loaded with NAGO. Okay, and so that led to the idea that Cyrano is an endogenous version of what had been seen in these viruses and artificial targets that the Stites and Zamor had found. That here the cells were making their own trigger, their assassin, if you will, of miR7, and is a very effective inhibitor or assassin. It's taking out 98% of miR7 in some of these tissues where miR7 is going up 50-fold when you lose the link RNA. Okay, so this showed that this process was also happening endogenously, and that cells were making their own triggers for TDMD to take out certain microRNAs. And in fact, this is one of the first examples of this. The other example, which was published a few months earlier, was from Elena Shkamutova's lab, and she had found that the NRAP RNA targets the degradation of miR29B. Okay, so Ben went on to show that this degradation, Cyrano-directed degradation of miR7 is part of this larger network of non-coding RNAs regulating each other in the mammalian brain, where you had these two microRNAs, the long non-coding RNA, as well as a circular RNA. And he had mice that really queried basically each node and edge of this network that he was able to characterize. And so that's caused us to be even more interested in Cyrano mediated degradation of miR7. And so Charlie, student lab, he started trying to find candidate genes that might be causing this, and he knocked down a bunch of those, but he really didn't see, and these were nucleases and terminal transferases and things like that, really didn't see any effects. And so he decided to do a CRISPR screen to try to find what proteins could be involved in this process. And that was successful, and he found that a key protein here is the Z-SWIM-H protein. And I'll mention that Josh Mendel's lab also did a CRISPR screen and also found that the Z-SWIM-H protein is required here. All right, so here's just some of the evidence from Charlie's paper. What he showed is that when he uses the CRISPR-I guide RNA that knocks down Cyrano in these K562 cells, you see about a 3-fold increase of miR7 and no real change in the loading control here. And at the same time, if you use a guide RNA, one of 2 different guide RNAs to Z-SWIM-8, he also sees a similar increase in miR7. And you see that Z-SWIM-8 is not required for this tailing and trimming, whereas Cyrano is, but Z-SWIM-8 is required for the destabilization of miR7. And when you looked more globally, here's just what happens when you knock down Cyrano. You see that you have this specific increase in miR7, and we see a similar increase and similar specificity actually in these K562 cells of when we knock down Z-SWIM-8. Okay, so really the only apparently the only highly regulated microRNA by this process in these cells. However, in other cells, you see many more microRNAs affected. Okay, so here we see in contact inhibited MEFs, Charlie was able to find 23 different microRNAs that went up. Okay, where in using cutoffs that did not identify any microRNAs going down. Okay, and this of course included miR7, but also in 503, which we heard about earlier, but in all the miR29, which we know about, but also another 20 that we didn't know about. Okay, and in fact, this is very interesting to us because we saw that these microRNAs that go up when you lose serino, I'm sorry, when you lose Z-swim8, are the same microRNAs that Elena had found had short half-lives when she was looking at their half-lives in wild-type cells. Okay, and in fact, when we look at the relationship between half-lives here and the sensitivity to loss of Z-swim8, we can see a really nice relationship. So what this is telling us is that the reason that these have short half-lives is because they're being targeted for degradation through Z-swim8. Okay, so that's contact-inhibited MEFs. Similar story in neurons. There, Charlie found 17 different microRNAs at his cutoffs, where again, nothing was going down at this cutoff, at his significance cutoff, and 11 of these were in neurons were the same as those in MEFs, but 6 of them were different. Okay, so it looks like this TDMD is really helping to shape microRNA levels, explaining the short half-lives of some microRNAs and kind of carving out expression domains of microRNAs in different cell types, presumably due to different expression of not only the microRNA, but also different transcription and accumulation of the triggers that target the microRNAs. And those could be expressed in different cell types, giving you different responses for different microRNAs. So really helping, again, to shape the levels of the microRNAs. We also found that this was also happening in Drosophila. So taking out the Z-swim8 ortholog in Drosophila, which we call DORA, you see 10 microRNAs go up, and this is the work of Elena Kingston again. And again, and these are the ones that are the Amara's lab had showed were the least stable in this cell type. And so we see that miR7 again is one of these substrates of the TDMD, but we don't see serino in these Drosophila cells. So presumably it's a different trigger in Drosophila that is causing the degradation of miR7. And a similar story in C. elegans from work, another graduate student, Michael Stubna. All right, so it looks like this pathway has been shaping microRNA levels really since the last common ancestor of mammals, flies, and worms. So what does Z-swim8 do? Well, when we did the lit search, this is what we got. Basically, nothing. This is not a highly studied protein, but on further investigation, we found a really great paper from Ishii Jin's lab at UC San Diego, and she had been working on the C. elegans ortholog of Z-swim8 and showed that it was a substrate receptor for a colon ring E3 ligase. And she had also done some work on the human ortholog showing that it was also one of these, part of this E3 ligase. All right, so an E3 ligase, okay, that's not a nuclease. And so that really required a revision of the model for TDMD. And so our lab, as well as the Mendel lab, came up with this model here involving this ubiquitin ligase. Just to get you oriented, normally you have microRNA-directed target degradation, okay, where you have the C pairing, recruiting the deadenylase, decapping decay of the mRNA. In this case, it's target-directed microRNA degradation. And the difference is that when you have this extensive pairing to the 3' end, that causes these conformational changes, which causes the tailing and trimming, but that actually turns out not to be on pathway, as both of our labs showed. And instead, what it causes is the recognition by this ubiquitin ligase. That then polyubiquitinates the Argonaute protein, which had been protecting that microRNA. And once the Argonaute protein gets destroyed, then you see degradation of the microRNA. In our labs, both Josh's lab and our lab, has a lot of evidence supporting this model. We found lysines on the surface of Argonaute that will inhibit, that when mutated to arginine, will abrogate this degradation. Proteasome inhibitors also prevent the microRNA degradation, and as well as various other lines of evidence. Actually, one of my favorites is this experiment that Elena did, and what she did is she used this tube reagent, which has affinity to polyubiquitinated proteins. And so she expressed this in the cells and then pulled on all the polyubiquitinated proteins and then sequenced the small RNAs. And she did it both in the Z-swim8 knockout cells, S2 cells in Drosophila, as well as in wild-type cells. And what she found was that preferentially in wild-type cells, the microRNAs that were associated with the polyubiquitinated proteins turned out to be the same microRNAs that were Z-swim8 substrates. Okay, and so this really showed that these microRNAs that are Z-swim8 substrates are associated with a polyubiquitinated protein, presumably the argonaute. And there's a really nice relationship between the extent that she would get this enrichment of these microRNAs in the tube enrichment versus their fold changes, the extent to which they were susceptible or substrates of Z-swim8 in the cell. All right, so we're quite excited about sort of our foothold now, our toehold into the this pathway of TDMD. We're really interested in using the knockout of this protein to find additional substrates of this process in additional cell types. We're also searching for the RNAs that are triggering the destruction of the microRNAs, presumably different triggers for each of these different TDMD substrates. Lots of interesting biochemistry to do here, and also the biology. It turns out that in flies, the Z-swim8 knockout is lethal. The embryos develop, but they can't hatch. Similarly, in mice, the pups are born, but they only if they're usually they die very soon, easily within the first couple hours. And so we want to know which dysregulated microRNAs are responsible for these phenotypes. And again, lots of interesting things to do in this pathway that has this very widespread and broad effect at shaping microRNA levels. So that was the microRNA story. Next, I want to turn, say a little bit more about microRNAs and how they regulate their targets, and then turn to mRNA post-transcriptional control. So just to zoom in on this part here, as I think most of you know, that what the microRNAs are doing is they're bringing argonaute to the messenger RNA, typically pairing to the 3' UTRs of the mRNAs, but you can have pairing elsewhere. And that then recruits a scaffolding protein, TNRC6, which recruits the deadenylase complexes, PAN2 and CCR4 NOD complex. And this shortens the poly-A tail. These complexes lead to poly-A tail shortening, which ultimately leads to decapping and decay of the mRNA. And on top of that, there's a little bit of translational repression, okay? But our lab and others have shown that this is only a small fraction of the regulation. So what we see is that over 2/3, often over 90% of the repression can be explained by this mRNA destabilization, okay? Just leaving a small fraction, detectable, but a small fraction happening at the level of translational repression. Now, this observation that it was primarily destabilization of the mRNA actually came to a surprise. Many people thought that microRNAs were primarily doing translational repression, but it actually turned out to be a relief for many people working on microRNAs, because what it means is that you can perturb the microRNA and then just look and see what's happening at the mRNA level and get a pretty accurate view of the effects of the microRNA. Okay, and so because it's a lot easier to look at mRNA levels than at ribosome protected fragments or protein changes, that turns out to be quite convenient. Unless you're looking in the early embryo, okay, it turns out that in the early embryo, as first found by Antonio Giraldez, if you look at changes in mRNA, you'll completely miss what the microRNAs are doing. Okay. There you have to look at ribosome footprint profiling. And I'll get back to that in a minute. So seeing that microRNAs having this effect on mRNA poly A tails, and that's how they're acting, led us to develop an assay for measuring the lengths of poly A tails. And so we developed a high throughput sequencing assay for doing this. Neri Kim's lab in parallel also developed an assay for this purpose. And these days we're kind of using a hybrid between the two. And what we found, and one of the first things we wanted to look at, is the relationship between poly A tail length and translational efficiency and the density of ribosomes on the mRNA as measured by ribosome footprint profiling. And so what we saw, and this is the work of Alex Subtolny and Stephen Eichhorn and others, what we saw is that in oocytes and early embryos, that there is this really strong relationship between the length of the tail and the density of ribosomes. Okay, longer tails, more efficient translation. And that was not a huge surprise because people had looked at individual genes in these systems, in the frog oocytes, in the fly embryos, and what they saw was that just looking at a few particular mRNAs, there's this phenomenon of cytoplasmic polyadenylation. It extends the length of the tail, and that causes a huge increase in the translation of that mRNA. And it's a way to regulate genes in a context where you don't have any transcription, all your mRNAs are inherited. So what our work showed was that this is not just happening to those few genes, it's happening more broadly, that this relationship between tail length and translational efficiency. The really surprising thing came later in development, where at the point of gastrulation, and then in all the post-embryonic tissues or the mammalian cell culture, yeast, we did not see any relationship between tail length and translation. Okay, so there's this developmental switch in translational control where at first translation is very sensitive to the length of the tail, and then that goes away. And we initially had seen that in fish and in frogs, and later our lab and Neri Kim's lab showed this to be true also in Drosophila. Okay, so we've been very interested in, well, first of all, this switch actually explained this mystery of why microRNAs seem to be doing one thing in early embryos and something else later in development. So let me just summarize that. So this is looking in zebrafish, and gastrulation is here. You have the early embryo, you add the microRNA, it associates here with the 3' UTR, that recruits the deadenylases, shortening the poly A tail. And in this system, shorter poly A tails lead to less translation, okay? With no real changes in mRNA abundance. Later in development, the microRNA binds, shortens the poly A tail, but in this system, the short-tailed mRNAs are degraded, okay? There's also some, translational repression, but this has nothing to do with tail length because there is no relationship between tail length and translation later in development. Okay, so the reason that you get these very different consequences of microRNAs is not because, the microRNAs are doing anything different, or not because of the dynamics of microRNAs or anything like that. In both cases, the microRNAs are causing the tails to get shorter. the difference is that the consequences of having a short tail is very different here than it is here. Here, if you have a short tail, translation is repressed. Here, if you have a short tail, the mRNA goes away. Okay, so what is it that causes this difference in the coupling between tail length and translation efficiency? Why do you have it early on, and then why does it go away? And so to address this, Coffee Xiang, a postdoc in the lab, has developed this reporter system and used it with oocyte extracts. And he has these two reporters, both identical except for their tail, okay? One has a short poly A tail of only 29 nucleotides. The other one has a tail of over 100 nucleotides. And when he adds it to this translation extract from frog oocytes, what he sees is this huge difference, okay? Clearly there's the longer tail mRNA is translated much better than the short. And he showed that it's not due to difference in stability or that the tail lengths are the same over the course of this assay. And so this really showed that there is a causal relationship here between tail length and translation. Not something that he saw in reticulocyte lysate, which is one of these post-embryonic systems that doesn't have strong coupling between tail length and TE. All right, so what then could be causing this difference? And one thought we had was, well, maybe it's polybinding protein. Okay, polybinding proteins normally thought to coat the tails of mRNAs, and maybe there's some mechanism that the cell would have to sort of count the number of polybinding proteins that are bound to an mRNA. Alternatively, polybinding protein could be limiting and involved in this process by just the fact that if it's limiting, the mRNAs with longer tails are going to outcompete the mRNAs with shorter tails for binding to polybinding protein, and thereby preferentially benefit from its boost in translation. Okay, so to investigate these possibilities, what Coffee did is he tried adding more polybinding protein. He just purified the polybinding protein, added it to the extract, and then looked to see what would happen. And there what he found is that as he added more polybinding protein, he found that the short-tailed mRNAs, compared to adding his control here, the EGFP, short-tailed mRNAs were translated better. Okay, no real difference in long-tail translation. Okay, so yeah, he saw that, and then that just started closing this gap here between the short and the long tail. He also did this by looking in embryos by first injecting more mRNA for polybinding protein or for his control, and then later adding the reporter assays, and he saw the same thing. Okay, in the embryo, this reporter's short-tailed reporter was translated much more efficiently if you have more polybinding protein. And then you could look at endogenous transcripts from these embryos just by using our poly tail length profiling method together with translational ribosome footprint profiling. And so in this plot here, each of these points shows the result for mRNAs from a different gene. Okay, so we're looking at about 6,000 different genes, mRNAs from 6,000 different genes. So you can see that those that have long mean poly tail length or median poly tail lengths, they tend to have higher translational efficiencies than those that have short. Okay, and this is what I was telling you before, this relationship between tail length and translation. And with a Spearman here of 0.61. Now, when he adds polybinding protein, or the mRNA for polybinding protein, and the oocytes start translating it, what he sees is this relationship goes down. Okay, now you're starting, it's only a Spearman of 0.36, and he sees the same thing for the embryonic version of that polybinding protein, that it goes down, that you no longer have a strong coupling between tail length and TE. Moreover, when he looked at the TE in the presence of additional polybinding protein compared to the control, that the in normalizing here to the TE of the mitochondrial mRNAs, which of course don't see any cytoplasmic polybinding protein, he sees that the translation of nearly mRNAs from nearly all the genes increases. Okay, and you can see that increase in translation just by looking at, you know, incorporation of S35. Adding more polybinding protein gives this really big boost in translation in this system. And most interestingly, it's the mRNAs that have short tails that increase in TE the most. Okay, whereas the mRNAs with longest tails really don't have any change. All right, and that's in fact what we saw with our reporters. Remember, it's the short-tailed mRNA that goes up when you add polybinding protein, not the long-tailed mRNA, and not controls. Like if you have a histone 3' end, no poly A tail, then you don't benefit from having extra polybinding protein, or the MALAT, this non-coding RNA 3' end that has no tail, no benefit from polybinding protein. All right, so what this is telling us then is that it looks like strong coupling between tail length and translational efficiency requires that the polybinding protein be limiting. And under these conditions, our model is that, well, the long-tailed mRNAs more effectively compete for the limiting polybinding protein. And this is especially going to be true if you can have cooperative binding of the polybinding protein. Okay, and then when coffee adds more poly A binding protein, okay, now it looks like these long-tailed mRNAs, they don't really change in their translational efficiency. So it looks like they have enough polybinding protein in this limiting system to get sort of maximal translation. Okay, but there's a huge difference for the short-tailed mRNAs. They're going from nothing to perhaps one polybinding protein, and that then allows them to benefit from polybinding protein. Okay, and so you see quite robust translation of these short-tailed mRNAs. Not quite as good as this. Okay, so there still might be some sort of coupling or a counting mechanism here, but still this difference between short-tailed and long-tailed really narrows. And so that's one really important feature of these coupled systems is limiting polybinding protein. So we know that that's required. One question then was, is it sufficient? Could we reduce the polybinding protein in an uncoupled system like, you know, mammalian cell culture and find that we could establish coupling? And so Coffey tried that using HeLa cells, and he knocked down polybinding protein in this experiment using siRNAs, okay, targeting PABC1, which is the dominant isoform in these cells, about 80%, and also PABC4, which makes up the other 20% of the polybinding protein in HeLa cells. So when Coffey just used the control siRNA, he gets what we typically see in these cancer cell lines and other cells growing in culture and other post-embryonic cells. Really no relationship between tail length and TE. Okay, the Spearman here of essentially 0. Okay, now when he reduces the polybinding protein to make it limiting, so he gets a little bit of an effect. Okay, the Spearman goes up a bit, but really no strong coupling between tail length and TE. So it looks like reducing polybinding protein, making it limiting, is not sufficient to establish coupling. Okay, and the reason for this actually comes from one observation here is that when you look at the tail lengths, after you reduce polybinding protein, they really increase to over 100 here compared to less than 80 here. Okay, so the tail lengths are increasing, and that turns out to be because the short-tailed mRNAs are destabilized when we reduce the polybinding protein. Okay, so here's the wild type pattern. This is an RNase H northern, just probing for the remnant of the the 3' UTR of GAP-DH after we cleave the mRNA. And that allows us to see the length of the poly A tail in these control cells. You can see that by the time you hit both of the poly A binding proteins and deplete them, most of the short tail and intermediate tail mRNA is gone. Okay, and you're left only with the long-tailed mRNA. And coffee sees this also using our more global techniques. It's just looking at all the genes in the transcriptome and the mRNAs from all those genes. And you can see that there's, especially for these short tails, these mRNAs that are, have tail lengths of 25 to 30 nucleotides long, you get this real striking depletion of the mRNA, and it extends also into this intermediate length, tail length mRNAs. Okay, so what this tells us is that, to have this strong coupling, we think that you're not only going to need to have limiting poly A binding protein, but when the poly A binding protein is limiting, the short-tailed mRNAs need to stick around. Okay, if they're not stable and they go away, two things happen. One is, well, you no longer have as many poly A binding sites, and so it's harder for poly A binding protein to be limiting. Okay, and secondly, if only the long-tailed mRNAs remain, then even if you have coupling between tail length and translational efficiency, if all the mRNAs are of a long, pretty consistent length, it's not really going to have an effect. Okay, so this is part of the reason why we can't establish coupling in these HeLa cells. But there's another reason. Oh, let me just say that Coffey went on to show that the reason that these short-tailed mRNAs are destabilized is because when they lose the poly binding protein, they're susceptible to polyuridylation. And so when Coffey knocks out these terminal uridyl transferases and you no longer have that polyuridylation, then the short-tailed mRNAs are stabilized. Okay, but that's not enough to establish coupling for another reason. And that's was revealed when Coffey looked at his ribosome footprint profiling experiments. And what he saw there is that in the presence of these siRNAs, which are knocking down the poly binding proteins, the mRNA, abundance goes down, and those distributions of fold decreases are just shown here for the mRNA. At the same time, the ribosome protected fragments also decrease, okay, pretty much the same, so that in the end, there's really no difference in TE. Okay, so when you lose the poly binding protein, there's actually very little effect on TE. Okay, so it looks like in these cells, poly binding protein has really negligible effect on translational efficiency. Very different from what I showed you. Remember that S35 experiment in the oocytes and all the mRNAs from all those genes going up when we added poly binding protein? Here, these cells seem to be insensitive. Their translation seems to be insensitive to poly binding protein. All right, so it looks like there are actually 3 criteria that you needed to establish this coupling between the length of the poly A tail and the efficiency of translation. First of all, the poly binding protein needs to be limiting. Okay. Secondly, the mRNAs that don't bind poly binding protein, they need to be stable. Okay, they can't just be degraded. And then thirdly, you need to be in a regime where poly binding protein actually enhances translation. Okay, if you're in HeLa cells and we think many other cell types, poly binding protein doesn't have any effect on translation. What it's doing is it's affecting the stability of the mRNA. Whereas here it doesn't affect the stability, it affects the translation. Okay, and so needing all 3 of these criteria to hold is the reason that we think it's been so hard for us to find contexts in which the length of the poly A tail influences translation. Okay, that they're just, it's the reason that only oocytes and early embryos is where we'd be able to see that coupling. Whereas the vast majority of contexts, at least one of these criteria are not met. Okay, and so in the broader take home from this is that, if you look at the textbooks, they'll tell you, okay, eukaryotic mRNAs, they have caps and they have poly A tails. And these are both required to stabilize the mRNA and enhance translation. And what I'm saying is, well, okay, this might be true for the cap, but for the poly A tail, we've been able to find some cells where the poly A tail and the poly A binding protein stabilize the mRNA. And we've been able to find other cells where these poly A binding protein and the poly A tail enhance translation. But we haven't been able to find yet a context where they're actually doing both. Okay, so it looks like it's either one or the other. Okay, and you might say, okay, well, that's just a minor change in the conjunction here, but this knowing, what the poly A tail is and what the poly A binding protein is doing in your cell type is really critical for understanding post-transcriptional gene control in whatever system you're looking at. And of course, I just want to thank then the lab. Here we are, current situation, and especially thank Charlie, Elena, and Ben for the work on the target-directed microRNA degradation, the TDMD, as well as help from Michael and Dan, and then Coffee, together with his predecessors, working on the role of the poly A tails and poly A binding protein. These people for funding, and others for all their help, and you for your attention.
Great. Thank you very much, Dave. Appreciate it. We have some questions in the chat, and if you have additional questions, please just type them into the chat, or feel free to unmute yourself and ask your question directly. So maybe while people are thinking, I'll start asking some of the questions from the chat. So one question from Adriano Bassini asks, do the levels of targets of microRNAs that are destabilized by TDMD also decrease in the Z-swim8 knockouts compared to wild type? So he's asking, what is the sort of cellular consequence of TDMD?
Yeah, exactly. So we certainly see that when we knock out Cyrano, we look at the targets of miR7. You can find some cells where and some cell types where, you can see a global de-repression, or I'm sorry, increased repression of the mRNA targets, the predicted mRNA targets. I have to say it's pretty subtle. You can see it when you look at a lot of targets simultaneously. You can get significance that way. It's clearly happening in the right direction if it happens. So yes, that is happening. In the Z-swim8 knockouts, we also see the same sort of thing. If we take it's a little bit more complex because you have multiple microRNAs changing, but if you focus only on the microRNAs that are changing the most and express the highest, we can see a signal for targets changing.
All right, thank you. And then Patnaj asks, how do the ribosomes get recruited differentially to mRNAs? For example, when the mRNA is a long poly A tail versus, I guess, the short poly A tail.
Right. So when the poly A tail matters, okay. So let's go back to here. When the places where the poly A tail matters, we think that it's actually this closed loop, which I know is quite controversial, but what we find is that when we add a poly A binding protein that cannot interact with eIF4G, then we do not see this enhancement of the short-tailed translation, and so so we think that it's the closed loop conformation that's helping to recruit, stabilizing the translational initiation factors here and helping to recruit the ribosomes in this context. Okay, here in these uncoupled systems, it doesn't seem like the closed loop is really doing anything, and people haven't really been able to observe it except for in a very few cases. And so, and in fact, when we use the poly binding or the polybinding protein that cannot bind 4G, it still stabilizes the mRNA. So you don't need to have this interaction for 4G for it to stabilize the mRNA. And here, it's not using polybinding protein. It's just being recruited, presumably by 4E and other factors, but has nothing to do with the tail in this context. Did I answer the question, do you think,
Yeah,
Frank?
I think that answers it. Yep. Yeah. Thanks. So Nancy Standard asks, presumably CCR4 NOT activity is low or non-existent in oocytes. I believe this is true of DCP1 and 2, but do you know anything about the CCR4?
Yeah. So I think you're asking why are the mRNAs with short tails stable in these oocytes? And that's right. The decapping machinery is not yet expressed, and it only is expressed later at gastrulation. And the same thing with these TUDases. So these terminal urtyral transferases that are important for destabilizing the mRNAs without polybinding protein in this system are not yet expressed here. So we think that both of those are the reason that mRNAs with short tails are able to survive in these oocytes.
Excellent. Gabrielle Taber calls into question what you just mentioned about the closed loop model in somatic cancer cells. So she asks if the differential effects of poly-A binding protein and the poly-A tails on translational efficiency could be perhaps due to closed loops forming only in certain cell lines.
Yeah, I think it would be, it's a great question. We haven't really looked at it directly. The closest we come again is just looking at these mutants that aren't able to bind to 4G. I think that part of the reason that this closed loop has been so mysterious and difficult is that I think it's really important in some contexts and not playing any role in others. And this idea that it could also be playing roles in other post-embryonic contexts, I think is quite fascinating. Be great to find those contexts. I'd be very interested in that.
Great. Do you have any questions, Sophia?
Yes, I have 2 questions. It's a very exciting topic, especially that will be very good to apply to cancer when it comes to cancer. So do you think that activated cells, because especially in hematological malignancies, we have like a high heterogeneity, and also we have quiescent cells and we have activated cells. Is there any way this tailing, shortening of the tail applies in different stages when cells are going through, for example, differentiation and kind of switching to quiescent to activated?
Yeah, we haven't looked at that. With respect to tail shortening, I think that's actually happening, everywhere.
Okay.
Except maybe not so much in the oocytes, but that we have looked at the dynamics of poly-A tail shortening, and that's a whole nother topic that I didn't talk about, but that is also quite fascinating. And you can see that some mRNAs, the tail lengths decrease 1,000 times faster than other mRNAs. Okay, so mRNAs from other genes, where for some it's it's about 2 nucleotides, 1 or 2 nucleotides per hour. For others, it's 30 nucleotides per minute. And that has a huge influence, of course, on mRNA stability. Anyway, I don't want to start talking about that other project, but just to say that the poly-A tail length and the shortening of poly-A tails is also an interesting topic. And what causes those big differences?
Exactly.
Still,
and a following up question is, and I'm assuming that the drugs we are using and the treatments we are using for cancer, they would affect this process, and how reversible this process is. Can we, is it manageable to kind of change the poly-A tail of the RNAs as we wish? For example, if drug is affecting, can we test whether or not drug treatment can affect the length of the RNAs, and based on that I'm imagining that we can even kind of manipulate the global gene expression in cancer?
Yeah, I mean, I think in most cancer cells, the tail length dynamics is primarily going to be influencing the stability of the mRNA, and to the extent that influence and slow down the shortening of the tail, that or somehow prevent the polyuridylation. I think those things could certainly influence gene control. Yeah.
Thank you. Yeah, because in cancer we know that most of the microRNAs in downregulated might be there is some connection as well there. So, thank you.
Great, thank you. Dave, I'm just going to ask one more question just to sort of sum up some of the other questions here. Somebody asks whether the COL2 ligase complex, you showed that it might have been involved in your paper, but does it phenocopy the increase in miR7? And then somebody else asks, how you think the other microRNAs are being affected by TDMD. Is it also through proteolysis of agos? And then lastly, just what are the general features that should allow you to predict which microRNAs are going to be subject to TDMD?
Okay, I'll start with the last question because I remember that one.
Okay.
So we do not yet have a way of predicting from sequence which microRNAs are going to be influenced by TDMD. So far, we think that any microRNA can be influenced provided that there's a trigger that's also expressed in that cell and that pairs to it with that extensive 3' pairing to trigger degradation of the microRNA. What was the other?
Yeah, the other question was any insights into the decay mechanisms of the bulk of microRNAs?
So what about the others? And that's a really great question, because what we see in general, and Elena did this in published it in her paper on the sort of microRNA half-lives metabolic labeling paper. She looked at the half-life of AGO by mass spec, SILAC, and she also compared that to the aggregate half-life of all the microRNAs and sort of the weighted half-life of all the microRNAs. And what she found was that is quite a bit longer. I think maybe about 3 times, I'm not quite sure, but about 3 times longer than the aggregate half-life of all the other microRNAs. So what that means is that for the long-lived microRNAs, most of them are not degrading due to degradation of AGO. Instead, the RNA is degraded and AGO is recycled to bind another microRNA. And so that's a very different mechanism than these short-lived microRNAs that are degrading through TDMD. And we don't understand that other mechanism at all.
And then I think the last question, actually, Chris is on the.
Yeah, I asked the Cullen question. I was just curious because in your work you show that the knockdown of the ZSWM8 or the elongin B and C increase the miR7.
Yeah.
But you also show that knockdown of COL3 increases miR7.
Yeah.
But the B and C make up the COL2 complex.
Yeah.
So
yeah.
What do you think? What do you think about that?
Well, I'd love to know what you think. This mystifies us. We think there's something really interesting happening here. Something very non-canonical is happening. And the Mendel group also had I think even better evidence. Those COL3, and so that's a surprise. And why would the elongins, which are only thought to be involved in colon 2 and 5, I think, why would they be critical? And then colon 3. So there's some really interesting E3 ligase biochemistry here that we're working on, but unfortunately I don't have the answers. But I agree, it's a very, very interesting question.
If you knock down the COL2, you don't see the same increase.
That's right.
Oh, that's really cool. Yeah.
Yeah.
Thank you. Because when I read that paper, we presented it for Journal Club. We were confused about.
Yeah. I mean, we didn't really emphasize this conundrum, but we're aware of it.
Thank you.
Okay, we have last question from Joanna. Joanna?
Hi, David. Thanks for the talk. So I had just a couple of quick questions. The first one is that I found it super interesting that you see the increase in ribosome binding in your long tail transcripts, but only in the embryonic stage. So I was wondering if you have any insights if ribosomal heterogeneity can play a role in this.
Right. So in fish, there actually is this sort of embryonic and adult form of the ribosome. We haven't looked at that at all, so we don't have any insight.
Okay, and then the second thing was a bit more technical very quickly, is that I noticed that you normalized all of your translation efficiency plots to mitochondrial transcripts.
Yes.
But I'm assuming that to look at those footprints, you used a different technique, right? Because those are going to be different, like the
They're different size, but you can use the same technique, and as long as you cut out the right size, everything works. Yeah.
Okay, I thought.
And the other thing to know is that, the ribo0 method of pulling out polyadenyl of pulling out ribosomal RNA for some reason depletes mitochondrial RNA. So we had to avoid using that, but that's kind of a technical thing that anyway, you can read about it in Coffey's preprint.
Yes. Thank you so much.
Yeah.
All right, I'd like to thank our speaker one more time. Thank you very much, David. It's been really great. And thanks everyone for listening. Thanks for your questions. And yeah, we will see everybody at our next event. So all the best until then.
Okay, thank you everybody. Great to see you, and sorry about the Zoom bombing.
Yeah, sorry about that. Bye.